channel biophysics, I was working in the more molecular level, okay, of organization. But now I tried in, no, you know, sometimes the funding is hard to get more translational science and to give a relationship between this beautiful channel with cancer disease, with most of the horrible disease in the planet. Okay, so before I start, I want to say something about our friends. Rafael was one of my best friends, was also one of the best friends of Eric. That's the reason why I meet Eric more than 20 years ago. So this guy developed the PhD degree with this. This is the thesis in Spanish. That is, I remember when Rafael told me that the future of the biophysics combining all our acknowledgement of ion channel biophysics with some disease, Carlos. That's the formula. That's the future of our field. So Rafael was a pioneer on this way. So this seminar is in honor of Rafael. Okay. So let me briefly introduce what we know about proton channel. The first report about proton channel was in 1972. 10 years after that was the first voltage line study. The people was focusing the physiological aspect of the proton channel. They are finding that the proton channel has a very tiny relationship with the immune system, in particular with and with some disease like asthma in the respiratory borne. But independent that many group was working with physiological carrying from proton channel, nobody, another part of research population was make a job behind us, this group that's the people that working with the ghost channel. But in 2006 was cloning the proton channel from 3 different species, from human, mouse, and C-square. C-square, by the way, is the cloning that we use in the lab. And the reason that we're using this clone is because this clone expressed very well in the oocyte. At the oocyte is the system that we use in the lab to express the channel. This is the heterologous system that we're using. Okay, so was cloning by 2 different groups, one in US, David Cladpan lab in Harvard University, another one is Okamura lab in Japan. And immediately the people was asked questions about more at molecular level, how the channels work, what is the selectivity of the channel, what is the permeation pathway, what is the stimulus that open the channel. The people that was working in Kv channel like me, very fast moving to this channel because this channel is a very voltage dependent channel. So here I show you in this slide the typical voltage dependent channel, the Kv channel, and you can divide these channels into functional domain. One, this is related with the port domain. Another one is related with the voltage sensor domain. So in comparison with Kv channel, that this is a tetramer, the proton channel is a dimer, and one of the subunities homology is equivalent to the voltage sensor domain in Kv channel. So that's the reason this is homology is more or less too high, this less than 20%. But anyway, the people that was working in Kv channel moved to proton channel and using the same technology, same methodology, same method that we're using for the study of Kv channel in the study of a proton channel. The other property of the proton channel is you remove the N and C terminus, you prevent in some way the dimerization process that you become this dimer in monomer. And the monomer is series function that you can see here. This is the microscopic current of proton channel that is faster in comparison with the wild type, with the dimer. The other property of the channel is this channel, as I said before, is pretty high voltage dependent, also depend of the pH concentration. If you increase the proton concentration, you increase the opening probability of the channel. The channel is also pre-selective to the proton, and you can modify, you can block the channel by the valent cation like zinc. And more specific with this is a specific blocker that developed in the last year, GBA. So we later on talking about this blocker. So regarding to the function of these channels, it appears in many tissues, like in respiratory, as I said before, in asthma, and during respiratory burn, also in sperma, in the move of the phagal, in the reproductive system, in the bone. Also, it has a very relation with almost all cells in the immune system. Of course, in the nervous system, in the glia, this is the immune system of the nervous system. And in the last year, have many reports about tumor cell that has a very important, very relevant relation with the proton channel. So today, my outline of my seminar is to convince you that the voltage-dependent voltage sensor domain is the pH sensor in Hv1 channel, and this one of the property of the cancer disease, the dramatic change of the pH together with another property like pain and like temperature that has a very tiny relationship with some ion channel, in particular pH dependency with proton channel. That's the reason we try to move in from this molecular level, from the voltage sensor, pH sensor, to the cancer disease. So let me start with the voltage sensor. Once the first question, this is how looks one subunit of proton channel, consisting in 4 transmembrane domains, S4, 2, 3, and 4. In the S4, you can find arginine. That's arginine is the candidate of the gating charge. Also, you can find the S4, the S0. This is intracellular segment that's in some way stabilize this protein on the membrane. So the question was, at that time, was where is the voltage sensor and how is work this voltage sensor? So in order to answer this question, we, for the simplicity, we become this dimer in monomer, as I said before, that we delete the N and C terminus. They keep mutant keep almost all property that the dimer, so that you can extrapolate all finding that you obtain in the monomer more or less to the dimer to see something about how work the channel. So we're focusing the S4 because the S4 have 3 arginines. That these 3 arginines was the first candidate to say something about the sensitivity of the voltage. Okay, so the idea was to using the accessibility experiment that we using many years ago in chest shaker potassium channel. So along S4 we put it cysteine, okay, and we with metanosulfonate reagent we test the accessibility of the position, and you can see here in just to show one of the position. This was 252. I hope that there was the position that here. So the idea was to test the accessibility of this position. 262 was 262, 264 in the open and the closed state. That's we proposed that it was one of the first models that how loose the proton channel on the membrane in the closed state and the open state. So with all experiment, this all kind of experiment, you can say something about the accessibility and the distribution of the amino acid residue on the membrane that have no idea what is the total gating charge that you're moving, what is the displacement, what is the mechanism, molecular mechanism behind of this displacement. So in order to answer how many charge, what is the total gating charge we're using, that the a method that we're using before for the Hv channel, this is a limit and slope that with method you can obtain the total gating charge related with the opening of the channel. Here I show you how looks this result for Checker potassium channel. As you can see, in Checker potassium channel have 4 subunits, 4 gating charge. The total gating charge, depending what is the distance that amino acid run by electrical field, you can obtain more or less that this is the number is 13. So we using the same strategy to obtain the total gating charge for the Hv1 channel. And you can see here, the total gating charge that was obtained at that time was 6, pretty convenient because we have 2 subunits, 3 charge multiplied 2 by 3 is 6. So we push the idea, we support the idea that these 3 arginines are important. These 3 arginines are gating charge during channel opening. Some of the reviewers, when we send the papers, wasn't in Nature Structural Molecular Biology papers, say that maybe you have contribution from the proton that move during channel activation, so you need to neutralize in some way. So that's the reason we made another publication in Journal of Physiology that we neutralize one by one the residues, the arginine that was in the S4, okay? And you can see here, the good news was when you neutralize, you replace the arginine by asparagine, you enormously decrease the total gating charge. The bad news was the decrease. You neutralize in one subunit, another subunit, you neutralize 2, 6 minus 2 is equal 4. That was less than 4. So with this result, we refine again the model proton channel. At that time, we more or less calculate what is the distribution of the gating charge and the interaction with another residues around of this arginine, and refines proposed for the open and the closed state the new model for the proton channel structure. So now the question was that these 3 charges are gating charges, but what is the displacement? What is the relationship between these both voltage sensor during channel opening? To answer this question, we're using one, this is special for you. I put it here, Eric. This is we're using the fluorescent, okay? This fluorescent, you put it one system close to the voltage sensor, the candidate of the sensitive voltage, okay? The move of the voltage sensor in this case was the S4. So you count more or less 5, 10 amino acids and put it one system, and you attach here the fluorophore. And the beautiful thing of this technique is you can at the same time to record the fluorescence as a result of the movement of the voltage sensor of this region, and also you can record the current as a result of the movement of the proton through the permission password. So with these 2 combinations, you can say something about the mechanism of the displacement of the voltage sensor by fluorescence. As you can see here, we published neuron that we find in this hook, and this hook was related with the state of the voltage sensor that converts in some way with permission password, with channel open. Okay, I don't want to spend many time with that, but the idea was that during channel activation, the S4 move from resting state to up state. When he's during this displacement, the permission pathway is opening, and in some way this movement of the voltage sensor converts with the permission pathway with the move from one side to another side the proton. Okay, so even that powerful of this technique that you can follow indirect in following the voltage sensor. We for many years tried to record the direct measurement of the movement of the voltage sensor, and that one is the gating current. We have a lot of experience in the lab with this measurement. Okay, here I show you in check potassium channel how looks the gating current, in calcium channel the gating current, in BK channel this is also potassium channel that has activation by calcium concentration also, and the BSP this is phosphatase that have voltage dependence. So as you can see here, this is 4 different patterns of gating current. At the gating current, you integrate this current, you can get the integration of the current is the charge. So you can obtain the total gating charge related with the movement of the voltage sensor directly. So in order to remove the macroscopic current and to detect just the gating current, we move it to this mutant. This is the in the last position here. I showed before there was 3 charts in the S4, but in the end part of this 4 is 1 asparagine. So we replace this asparagine by arginine. As a result of this electrostatic effect of the arginine, we remove a lot the macroscopic current, but we keep intact the displacement of the voltage sensor. You can see here from the fluorescent signal. Okay, so using this mutant, we start to study the gating current as directive movement of the voltage sensor. As you can see here, this is the first component as the gating current. We call Q ohm. This is the current ohm. And you can see here, you have the first movement of the voltage sensor, then continue with the macroscopic current. This is impossible to abolish completely the macroscopic current as a result of movement of the proton from one side to another side. But the phenomenon that we saw immediately was that the off component was disappear. So it means that the gating charge that you move in one direction never come back in this time to the resting position. So the question was, how is the displacement? How many kinetic state is necessary to move the voltage sensor? That we published recently in PNAS that we need at least 5 kinetic state for the movement of the voltage sensor, distinguished clearly. 3 of them very fast movement of the charge, and the last 2 is more slower. So that's the most convincing model, kinetic model for the displacement of the voltage sensor. And with this kinetic model, we mimic the same result that we obtained experimentally. Was keep it the question that what happened with the off component? What you need to wait for more time to recover the gating charge? So the first question that we asked was this effect, this phenomena is belong to in general to the channel, or this property is belong to the mutant that as a result this replace of this asparagine by arginine. So in order to answer the question, we replace again to this arginine by asparagine, and we move it to the position aspartase 116. This aspartase, from another group, and Tom DeCourcy group, they propose that this is the selectivity filter, one of important residue to the selectivity filter. So he just replaced this aspartase by asparagine. You recover the off component, you can see here. So this means that these properties belong to the mutant and not to the wild type channel. So in order to establish the same effect, we make a double mutant. As you can see here, the good news was that they disappear. Okay, it's make it slower that in comparison with the previous mutant, but was not too much like the first one, like when you have just the replay for the position 264 for one arginine. So how you can explain this phenomena, okay? So this is the structure, Hv1 channel, and here you can see in the S4 you have 3 arginine, this position 255 to 261, and the last one, this asparagine, okay? If you replace this asparagine by arginine, in some way, you induce the interaction of these 3 or 4 charges with the aspartate in the selectivity filter. You can keep the voltage sensor in the up state. So with this arginine, you repulse the asparagine that you have around of this charge, okay? In some way induce this interaction between these 2 arginines by this aspartate and keep in the up state of this channel, the voltage sensor. That's the reason why that the off component disappear, and you need to wait for more time to recover the gating charge to back to the resting to the resting state. So now we figured out more or less what is the voltage sensor, how it works the voltage sensor. But I want to convince you that the voltage sensor in Hv1 channel is a pH sensor. So to study that one, we the voltage sensor is the pH sensor is dependent the delta pH. Okay, we calculate the delta pH by pH intracellular, and in the intracellular side means the extracellular side. This is the definition of the delta pH. Okay, so the idea was to using the mutant that related with the selectivity filter, because we completely abolished the macroscopic current, we keep intact the gating current. I showed before, okay, as you can see here, this is the on and the off component intact. So it means that the chart that you move in one direction, going back perfect to another direction, okay? So using this mutant, we explored, okay, the delta pH. As you can see here, when you decrease the proton concentration, okay, you make slower the kinetic of the gating current. But as you can see here, you put it the pH 6 in the intracellular, in the extracellular side, 7 in the extracellular side. Delta pH is equal 0. And when you have the equal 0 pH, you have the perfect overlap of the QV. So it means this is the chart that you move during movement of the voltage sensor. Okay, as you can see here, the delta pH is important in the voltage dependent for the gating current. So as you can see here, this is the perfect overlap for all delta pH equals 0, independent what is the absolute pH that you put it in the extracellular and extracellular part of the in the pipette or in the bath of the experiment, during the experiment. Okay, as you can see here, when you make the delta pH 1, you shift the curve to the left, and you keep delta pH 1, you have again the overlap and the curve. Okay, and then you make delta pH 2, and you achieve more the curve. So it means that for the gating charge is important, for the voltage sensor is important the delta pH, and the delta pH is the response of the in some way of the movement of the voltage sensor. So in other words, the voltage sensor and the pH sensor is the same domain. So the same result we obtained before for the GB. So it means the DGB relationship between GB is a result of the movement from one side to another side, the proton. This is the more relation with the opening of the channel. And this opening the channel is also the pH dependent, delta pH dependent, like voltage sensor dependent. So how to explain that phenomena? So here, so you have the delta pH for the voltage sensor, that this means the pH sensor, but at the same time you have here the permission password. So that's the conclusion that we did here. We have in the same region, three processes: the voltage dependent by voltage sensor, the pH dependent by voltage sensor again, and also the permeation pathway. When you move the voltage sensor as a result of the polarization, you in some way dissipate the gradient that you have by the difference of the pH. That's the conclusion that we did, that just to dissipate this gradient, that's the reason that the voltage sensor and pH sensor depend on the delta pH, because has a very tiny, in the same region, the permeation pathway. That's the reason also the permeation pathway has. And in particular, in the end of this part, you have a very important amount of hydrophobic amino acid residues that keep a lot of molecular of water, that from this molecular water jump the proton to move it through one side to another side. So this is the at the molecular level, that conclusion that we did, we learned for many paper publication years about how it's work, proton channel, voltage sensor, and pH sensor. But what is the relationship with the cancer? So let me start with tumor cell. The tumor cell, once the property of the tumor cell is have a very high metabolism, pyruvate metabolism. As a result of this, pretty high in comparison with another cell, they produce enormous amount of proton that need to release to extrude outside of the cell, because the cell doesn't like a proton concentration. So by proton channel, by Hv1 channel, you can relieve this enormous amount of proton concentration as a result of this pyruvate metabolism, pretty high metabolism that you have in cancer cell. Even if you one of the you want to stop the growing of the tumor, you can stop to eat carbohydrates. Okay, all sugar that you eat in some way increase the proliferation of the tumor. So the idea was, if you block this channel in some way, you keep a very high concentration of the proton here, you can kill the tumor. What's on the strategy? But one of the problems with this strategy is that this tumor cell can live in very low pH, low value or very high proton concentrations. You measure, the physiological measurement of the pH is around 7.2, 7.4. So this cell can keep alive in PS6, 5. So this becoming the very, it's like a trap, okay? This increase the proton concentration around of that, like to defend from the killer, like T cell. So now the question was, what I doing this Hv1 channel in another cell that more related with the microenvironment of the tumor. So here I show you the tumor, and you have all other kinds of cells, T cell, dendritic cell, and this is macrophage, and also you have this myeloderivate suppressor cell. This is sort of the differentiate cell that this cell is pretty important in the immunosuppression process of the T cell. So in other words, when you have some inflammation process, you have tumor or whatever, cut, I don't know, different signal induce the proliferation of T cell. That you need some suppression system that stop the T cell, and this is the myeloderivate suppressor cell. This suppressor cell has two markers. This is how we identify this suppressor cell, CD11 and GR1. And by these four mechanisms, they can make an immunosuppression of the T cell by system arginine depletion, by damage of the viability of T cell during traffic of this kind of the cell, during proliferation also. And one of the most important is the production of the ROS and the nitrogen species. So we are focused in this mechanism, how the most important mechanism during immunosuppression of this kind of the cell, called this myeloid cell during cancer disease. This is related with the solid cancer. Okay, so the idea is that when you have a tumor, you have a lot of T cell, and this T cell has modulated in some way by myeloid derivate suppressor cell. So what about the Hv1 channel relation with the Hv1 channel with the immunosal? So here I show you eosinophil, dendritic cell, macrophage. Has all these immunos cell have a lot of a very important amount of Hv1 like protein, okay? That they these proteins combining with this complex of the protein, NOX2, okay? They together have a mechanism by which macrophage, for example, defend the organism of the bacteria. So the question here is, in the myeloid cell that is this cell is important in the microenvironment, is combining with the NOx, and it's also Hv1 channels appear in this kind of the cell. So we detect that the activity of the NOx is pretty high, and the reason that why these all the species are together working. So we immediately try to finding some or identify some presence of the Hv1 channel. So in order to detect the presence of the Hv1 channel in this kind of the cell, we made a culture of myeloid cell. Okay, here I show you the maximum of this is around 4 days, 96 hours. You can obtain the 80–90% of the cells, like for the both market for the GR1 and CD11, as identified that this is the amyloid superficial cell. So in this condition, with this cell, was the first experiment that we did. This is a monomer of proton channel. The weight of the monomer of proton channel is around 330 kilodalton. We obtained the very beautiful band for 30 kilodalton. So was the first finding, okay, that the proton channel is in this kind of the cell. Nobody report that one. So also using the cytometry, we also detect that in this cell, almost in 100% of this cell have the proton channel. So by microscopy, we identify what kind of cell we want to record to ask or to answer the question, this Hv1 channel that appear in this cell is functional or not. Okay, and you can see here, for this cell, this is the myeloid cell. This looks more than the dendritic cell, and this clear that the marker, this is the marker, that the marker for this cell that identified this cell like a myeloid superficial cell. We record essentially this kind of cell, more sickle, okay, by patch clamp, and we obtain a beautiful, clear macroscopic current that belong to the proton channel, Hv1 channel. As you can see here in the end of the trace of the macroscopic current, you can see the decrease in the current. This is the typical phenomenon of proton channel. This is no inactivation, no blocker. This is the depletion of the proton. This is, you can see here, this is different pattern of proton channel from different tissues, from different cells, in the sperma, for example, that this phenomenon is pretty clear for proton channel. And you can see here, if you change the length of the pulse, you decrease for more high polarization pulse, the length of the pulse, so you prevent some way this phenomenon. You prevent the depletion of the proton, and you keep intact the amplitude of the macroscopic current. That this phenomenon belong to the proton channel, because it's impossible with this change to obtain this result for the inactivation or whatever blockage you have in the preparation. Okay, so using this kind of the protocol, we make a characterization of biophysical property of the proton channel from the myeloid cell. Okay, detect the selectivity for the proton. You can see here it's pretty clear. So also we count the channels. So using non-stationary noise analysis, we obtain that you have around 9 channels, okay, in this kind of the cell in the membrane. You have a lot of amount of proton channels. So the question now is, what's going with all these channel in this cell? So in order to answer that question was that the role of Hv1 channel, we're using 2 blockers. One is zinc that we time ago published the paper that describe more or less the molecular mechanisms of the blocking by zinc. This is 2 in the extracellular side. You have 2 sides, okay? One more extracellular, one less, okay? And you can see here, independent what is the concentration you're using, you can block the macroscopic current by extracellular side, and using total different protocol, more like a guanidine derivative. This is the GBI that previously I mentioned. Okay, this blocker is more specific for the proton channel, and you can prevent the macroscopic current by the intracellular side. So Now you have two completely, biochemist point of view, different blocker, and you can using this strategy to, in some way, explore it, to study what is the function of this Hv1 channel in this myeloid cell. So the idea was to test this molecular mechanism. We know that the NOx, during NOx activation, you increase the proton concentration in the intracellular side. As a result of that, this concentration, you polarize the membrane in some way, and you increase the opening of the channel, the probability of the channel to be open. In some way, also, you increase the proton concentration. These two factors, voltage depolarization and the increase of the proton concentration, induce the increase the open probability of the channel. As a result of that, you open the channel, okay, and you have the feedback between these two species, and you can relieve from to the extracellular side the proton, and these protons combining with the oxygen, singlet oxygen, and as a result of that, you have peroxide. The peroxide is the responsible of to kill the bacteria. That's more or less the mechanism, that canonic mechanism that you have for the this combination between proton channel and NOx2. So in order to explore this, what is this interaction in following, our idea was to following the production of the ROS. Okay, we're using this fluorescent probe. I don't want to spend time, but the finding was that here, when you're using zinc or more specific blocker, GBI, as you can see here, you decrease the production of the ROS. So it means that that's the mechanism, that one of the possible mechanisms by which the Hv1 channel, okay, in some way modulate the T cell. Is this true? It means that you block the channel, you block this interaction, this feedback. As a result of that, you reduce, okay, the production of the ROS. As a result of that, you keep free the T cell, and the T cell is free to kill the tumor. So in order to test what is the relationship between this cell, in particular with Hv1 channel with T cell, we made a proliferation test. So we take a splenocyte culture, and this is the marker for the splenocytes, almost 100% of splenocyte, and we use it con cannabalin A, like in mitogen, like to induce the proliferation of this T cell, okay? And the idea was this is empty myeloid cell, okay, combining with T cell in the same culture, okay, and now we induce proliferation of the T cell by this mitogen concanavalin A, and then when you block the proton channel here in the myeloid cell, you in some way prevent the production, decrease the production of the proliferation of T cell. So in some way you affect this part. As a result of this blocker, you affect the T cell proliferation. We obtain, okay, the same result for the both, for the zinc and for the more specific blocker. That we test 2 hours, 12, 1 day after blocker incubation of myeloid cell with before we mix with T cell to test the proliferation of this kind of cell. So now the idea was, what is the relation with the tumor? Okay, because everything that we show here was in vitro. What happened in vivo? What's going on with the real cancer? So using the animal vivo, okay, the mouse, we induce the melanoma of this mouse. Okay, as you can see here, clear the tumor. Okay, we measured the tumor in order to uh, test what is the possible strategy, therapeutic strategy for this kind of the tumor by blocker of Hv1 channel. And you can see here, this is how looks how increase the volume of the tumor and how the decrease. Okay, how's the mice pass away pretty fast in almost 35 days. All population of the mice disappear. Okay, and here I show you the migration of the myeloid cell from the bone marrow to the spleen. So as you can see here, this is decrease in the bone marrow and increase in the spleen as a result of migration from one region to another region. And if you combine this tumor with some reagent to block the Hv1 channel, specific Hv1 channel, you can see the normal effect of the decrease of the tumor. This is by treatment, by this therapeutic strategy. Okay, you block the Hv1 channel, you significantly reduce the tumor volume. As you can see here, this is how it looks when we make a prediction for more days to keep this the strategy. You increase the concentration of the reagent to block the Hv1 channel. You amazing the reduction of the tumor for almost nothing in the mice population. So in conclusion, we can say that the Hv1 channel that appear in the tumor cell, you can block it, all these Hv1 channel. You put it, the blocker, specific blocker for Hv1 channel, you can kill the tumor, but at the same time, you can reduce the immunosuppression of the myeloid cell to the T cell, and the T cell has less concentration of proton channels. That's the reason keep alive to be possible to in nature way to kill, to destroy the tumor. That's the idea of this therapy, where the Hv1 channel is one that's important target, combining with another inhibition that you can use during cancer therapy. So before I finish with my seminar, I would like to say thanks to my group, to my lab, in particular, Juanjo, Miguel, Antonio, and Christian and Juanjo in particular, developed this all a very important portion of this research. And also my college in Chile, Osvaldo, Alan, and in particular the Millennium team, and my co-worker in USA, Peter Larson, Luis Cuello, Héctor Baraja, and Mexico, Angelica, and in Argentina, the girls, Veronica Milesi, Gabriela Modeo, and also, of course, Marcelo. Before I finish this presentation, I would like to say that 16 minutes ago, maybe more than 1 hour, we already have the publication of this part of my presentation in Pena's paper. Thank you so much.
Thank you very much, Carlos. That was an amazing presentation. We have actually some time for question. Does anyone has ask a question to Carlos?
Yes, I would.
Oh, okay.
Okay, you talked about, and we talked about earlier, about the disappearance of the, in terms of the basic properties of the channel, the disappearance of the return of the charge under the control conditions. Did you ever look at, see how long the depolarization or the depolarization pulse has to be before you initiate that response? How fast does that phenomenon occur?
Okay, let's thank you, Martin, for this. Which it's not real disappear. You have
Right.
you make a slow with this. No, the idea is with this mutation. This is not a real trapping. So we for I don't know 3 or 4 days was discussing that. Was a very hard discussion with the people that was participating in the publication, that how to call this one. This is not a real trapping. This is not a real trapping because if you wait at least for millisecond, okay, you can recover completely the gating charge. So you apply the recover from, it's like inactivation process in check potassium channel. So this is not a mobilization. This is not trapping of the chart. You need to wait for more time to destroy the interaction between aspartate that you have there in the S2 with the arginine that you have in the S4. As a result, you keep more time with this mutant in the up state of the voltage sensor, okay, for more time. But you wait for at least millisecond, you can recover again the same state of the voltage sensor. So you can recover completely the gating chart that you move in one direction to another direction. So this is not the real trapping. It's not the immobilization process. It's just to wait for more time, more microseconds, milliseconds, the recovery from this state, the voltage sensor domain. That's the only thing. So we have a thesis from Miguel. Miguel have here the thesis. I show you here. I put it. I know that Marty loves this kind of so I show you, Martin. Let me see. I try to. Let me see. Try to figure out permission. Miguel. Let me see. Miguel, I can't find. Let me see. Miguel presentation, CPPT. Okay, let me see here. This is the Miguel thesis. As you can see here, this is the phenomenon of the trapping. I don't know if you can see that one.
It's hard to see, Carlos. You might need to zoom in a little bit. There we go.
Yeah?
Yep.
Nice, okay.
Even more.
So, Now it's okay.
Yes.
Let me start with that. Okay, you put it here in the play of this asparagine, one arginine, you get this phenomenon that Martin was talking about. Okay, this is the some people call that one the like immobilization charge, and other people like it trapping.
Okay, can I ask a question right here? What
Yeah.
if you turn this one off right at the beginning? You see the full current coming back?
Yeah.
Okay.
That's
how fast does that set in?
This is Miguel, help me with that. Exactly, what is the values in 5 milliseconds? Yeah, more or less 10 milliseconds. You can, if you wait for, you make a recovery from inactivation or from, this is like a recovery from inactivation, okay? It's sort of inactivation process, okay? You wait for 10 milliseconds, you can recover completely the off component of the gating current. So here I show you, or we show, or Miguel showed the molecular mechanism here. You can see here, this is the trapping, you know, the trapping, okay? And you can see here, if you replace, okay, this asparagine by this arginine by asparagine, you recover the off component, like you can show here. And if you change the hydrophobicity of this interaction also, so you have two driving force here, the hydrophobicity and the electrostatic interaction. In other words, you keep in the up state, this is a molecular dynamic, in the up state, the voltage sensor. You induce in some way, stimulate in some way, this interaction between this arginine with this aspartate, and you keep for more time the voltage sensor in the up state. So you need to wait for at least 10 milliseconds to recover the voltage sensor to the initial position, to the resting state. That's the explanation that we did for that, but it's clear. So we already make a manuscript. We very soon, I think that the next week, we submitted this, how to explain this, because this phenomenon is pretty common in this trapping in many channels. Some people call demoralization, another trapping, but I think that now we have like a very molecular explanation that what's going on, in particular in Hv1 channel, with this how it's worked, the voltage sensor relationship with
the trapping might be or the mobilization mechanism might be different for sodium channels, for example.
Yeah, maybe it's different. So I need to, but I'm not pretty sure. You need to find what is the driving force that govern this mechanism. We find that you have the electrostatic, but not only. The hydrophobicity is pretty important also. The environment around of this is pretty important to keep in one position, to keep to frisk the voltage sensor domain. That's the idea. So
are there any other questions? If not, I would like to ask a couple of questions to you, Carlos.
But you can alone. We can.
Yes. So these proton channels are these proton channel currents are basically omega currents.
This is pretty important question. So this is not the real omega question, the real omega current. Okay, omega current is the when we try or figure out what's going on with the permeation pathway, we push the idea that this is sadly like omega current in Check potassium channel, because in Check potassium channel you have two currents. Okay, one that belong to the pore domain. Okay, this is the ionic current, potassium that move from one side to another side. But also you have the current in the S4. So this is sort of omega current, but it's not, it's sort of omega current. You replace the last arginine, the R4 in Check potassium channel, you can become in proton channel. This is the Check potassium channel. So he worked like proton channel. So this is the point that you want to address here. So if you replace, look at that, you have in the S4 of Check potassium channel, you have 4 charges: 362, 365, 368, and 371. 371, you replace this arginine by histidine, you become this channel in proton transporter. That is one of the papers that did Dorin Starace with Pancho Besanilla, that they demonstrate that the gating, the omega current, the first report about omega current was from Issacar lab, from, by the way, Peter Larson was the first that record omega current. All this idea passed to Tombola, to Francesco Tombola, and they published a beautiful paper about omega current. But Pancho with Dorin Starace, what was at that time his postdoc, he demonstrated that you replace this last charge, you become the channel in proton transporter. And this is pretty important because when you modify this asparagine, that this is equivalent position in Check potassium channel, if you modify this asparagine by arginine, you in some way modify not only the selectivity, you use also the number of the single channel by non-stationary noise analysis, we detect, or we explored the conductance, single conductance, because it's pretty low. It's impossible to record the gating record in Hv1 channel. So by using non-stationary noise analysis, we following the single conductor. So if you modify this equivalent position, this is N264, by arginine, you modify in some way the conductance of the channel and also the selectivity. So you can push the idea that this is the gate of the channel, that this is a possible gate that by which the proton go through the protein. That's the idea that you can know.
Great, thank you. That. My last question is that, do you know if these proton channels are expressed in skeletal muscle, like or cardiac cells? And if though, if
we can collaborate. We can collaborate.
What would be the function?
Okay. I don't know. No. Okay. Proton channel is has a lot of relationship with the, of course, proton concentration, but also you can combine this role with another. This is a transporter. Okay, it's proton transporter. It's also pretty important, and it's another species that's important in the pH control. So, but the proton channel is pretty studied in whole immune system. That I don't know, is because only in the immune system is important the pH values or the proton concentration, or it's because the people that was working in immune system was lucky to find the Hv1 channel. But every year appear in more important functions related with another tissues, like in the sperma. It's pretty important. The movement, for example, Hv1 channel is the most important protein in the movement of the of the spermatocytes. It's pretty important. You modify that one, you modify the fertility with the of the organism. Okay, that's but I know pretty sure that what is the function in the skeletal muscle. I think
Okay.
that's pretty important, maybe with the lactic acid. I don't know, it's during exercise. Okay, we'll see, because it's too many dramatic. You know, the problem is in the immune system is pretty important, not only the pain, only the temperature, also the proton concentration. So that's the reason the nature put it there, proton channel. Okay, so it's important. In muscle, I know pretty sure. I don't want to be honest. I don't know.
Okay.
Okay,
Thank you.
but we can collaborate. We can find what is
Yeah.
the present and the role of the Hv1 channel.
Yes, that would be a nice way to
A nice, okay.
Yep. Are there any other questions from the audience?
Not too many people.
Yeah.
Thank you. Thank you so
One last
much.
question, Carlos. Carlos, one last question. In terms of the drugs that you use, these blockers for the proton channel, are how specific are they, and are there any clinical trials using similar drugs currently?
Okay. The answer is yes. So we try already the clinical, because you know the cancer is, in comparison with another disease, asthma or diabetes or whatever, is you have a direct relation with the death. Okay, you have cancer, and in particular the people have this problem in the last stage of the cancer. You have problem with the therapy, because and there is pretty important, the Hv1 channel. It's true in the last state of the disease, that you can see enormous amount of the proton channel. Even this Hv1 channel is pretty important with the metastasis process. Okay, so let me show you one here presentation. Okay, so this is pretty exciting finding in the lab.
Yeah, indeed, it's very remarkable the effects that
okay, let me show you.
Have on the
so you can use proton channel not only like a therapy in the last stage of the cancer. This is only for the solid tumor. Okay, for the solid tumor. Okay, when the people have cancer in the last stage, they almost lost all function in the body. You have no hope to solve the problem with that. So one of the more protagonist role of the protein that relating with the cancer is in the last stage of the cancer is Hv1 channel, and he also is pretty important in the metastasis. And you can see here, I show you two different line. One more related with HeLa cell line that you can see is enormous amount of proton channel in comparison with another kind of the cancer that have it's not pretty aggressive, it's no make a metastasis at all. Okay, this kind of the cell that the people even using like a system to express the protein to study ion channel. Okay, this kind of the cancer is pretty metastatic, so you can follow it the possibilities of the metastasis of your cancer following the expression of Hv1 channel. This is pretty important because, you know, we have now a problem with the detection system. The immune system has reaction when you have a very some peptide with what is the length the peptide, the immune system react at this like orange, okay, and immediately activate all the mechanisms to kill the peptide. But sometimes you have the problem because you have a very small peptide that around this nucleate the tumor, as it's impossible to detect this peptide. And when you using all, I don't know, technique or therapy that you're using to kill, to abolish the cancer. However, you can detect that in this condition, the expression of the Hv1 channel. You have a very high expression of Hv1 channel. It's a very good marker that you for sure have the metastasis in the future. So you can using this Hv1 is like not only the new therapy for the to kill the cancer, and also to following, okay, like a marker to the metastasis. I don't know if I answer this question. I don't want to talk too much about that. So I want to say that we have here, okay, before I finish, let me show here that we have our group, the that's, let me, right here. This is the Milenio team. The director of this project is, this is a very, I want to spend a little time with that because this is a very good combination of different formation. This is quantum chemistry, this is quantum, this is engineer people, and this guy is more technology, engineer technology. But together with me, we working in to in some way once the line to using the first stage to understand the nanotechnology in nano field. What is the rule that government behind of this field? And then using nanotechnology like the environment of the drug to relieve, okay, in the right place, the reagent against the cancer. This is point of view is pretty important because now it's not pretty important what is the affinity of the reagent, how directly, what is the affinity of dissociation constant that you have, what is the concentration you need to use. Using nanotechnology, you can for sure to relieve in the right place, independent, to using the right administration. This is another strategy with this team and with this behind of this project. One of the line in the future, not now, is to combine our funding with the reagent with the nanotechnology. These two, all these people have a lot of experience in nanotechnology. These two, Valeria and Natalia, have a lot of experience how to make nanoparticle. My dream is to combine all these findings of their experience with this drug and to, in some way, to prevent the solid tumor in the future. That's the only I can to say about this group. I would like to thank also the rest of the college that helped me with this project, in particular my student.
It sounds very promising, Carlos. Thank you very much for the great presentation. Thank you all for attending.
Okay, bye-bye. Thank you so much, everyone.
Carlos, if you want, we can stay a little bit if you want to keep
Yeah.
to
Yeah.
talk